Welcome back students to this section of MCQs. So let us go further. All of the following metabolic pathways are active in the liver during starvation except. So we are talking of two things here. One is starvation and the other is liver. So be careful of what your answer could be. Look at both the conditions, liver and you are talking of starvation. So let's see what is the answer. Synthesis of VLDL. Other than this, all other pathways are active. So ketogenesis, liver is the only site for the synthesis of ketone bodies. So naturally, ketone bodies synthesis is going to take place. Glycogenolysis is the first few hours of starvation. The main way by which liver maintains blood glucose level is by glycogenolysis of whatever stored glycogen is there. It undergoes glycogenolysis and maintains the blood glucose level. HMP shunt is not related to fasting, but whatever it is, the liver needs HMP shunt. Why? Whatever NADPH is produced, it is kept on used for the maintaining the cell membrane integrity and for acting as an antioxidant and all this. The liver needs HMP shunt to produce NADPH, to produce the nucleic ribose 5-phosphate, the pentoses. So it requires HMP shunt that will be kept active. Gluconeogenesis is definitely occurring as soon as the glycogen stores are declining. Gluconeogenesis is going to take place. So first initially it will be glycogen which will be giving the blood glucose level. Gluconeogenesis will be low first, but as this is increasing, gluconeogenesis is going to increase. So this will be glycogenolysis and this will be gluconeogenesis. So this is how they both will help in maintaining the blood glucose levels. What about synthesis of VLDL? What is the main function of VLDL? Transport of endogenous triacylglycerol. So endogenous means triacylglycerol has to be synthesized in the liver. So for the synthesis of triacylglycerol, what is required? Fatty acid synthesis has to take place. And for fatty acid synthesis, what is required? We require enough fuel stores. Only then there will be synthesis of fatty acid. De novo synthesis of fatty acids occurs when the acetyl-CoA levels are adequate, ATP levels are adequate. Only then de novo synthesis of fatty acid occurs. So when that is not happening, triacylglycerol synthesis will not take place and synthesis of VLDL actually decreases during starvation. So synthesis of all other metabolic pathways are active except synthesis of VLDL. We'll go to the next question. The enzyme absent in liver. So did you get the answer? The enzyme is succinate thiophorase. You might have got it confused with succinate thiokinase. Succinate thiokinase is an enzyme which is involved in substrate level phosphorylation in the TCR cycle, whereas succinate thiophorase is an enzyme which is responsible for activation of ketone bodies. So you have got acetoacetate, ketone bodies, which may be got from beta hydroxybutyrate. Beta hydroxybutyrate can form, depending upon the state with an NAD plus balance or whatever it is, beta hydroxybutyrate dehydrogenase, depending upon NADH to NAD plus ratio, it gets converted to acetoacetate. Acetoacetate for its further utilization needs to be activated. So in this we have got a special enzyme in which it combines with succinyl-CoA. Succinyl-CoA gives its CoA to acetoacetate and converts it to acetoacetyl-CoA. Acetoacetyl-CoA can then be broken down to 2-acetyl-CoA, which enters the TCA cycle. So this enzyme which activates acetoacetate using succinate is called as succinate thiophorase. Succinate thiophorase. Now, where is all this taking place? This is ketolysis, and where ketolysis always takes place in extrahepatic tissues and not in the liver. So liver most of the pathways occur in the liver except ketolysis. Ketolysis cannot take place. It is only a site for synthesis of ketone bodies which are water soluble, alternative fuel to all other tissues like brain, skeletal muscle, adipose tissue. It sees to that all other tissues get the required fuel at whatever times it is required. by synthesizing ketone bodies, but it does not utilize ketone bodies. So it spares the glucose, muscle protein degradation for the and forming gluconeogenesis forms glucose. So whatever glucose that is formed from gluconeogenesis, let all the tissues which are absolutely dependent on glucose, let them use it. Most of the other tissues will shift over to, including the brain, will shift over to ketolysis during starvation. So the enzyme deficient absent in liver, pyruvate carboxylase, no, because it is gluconeogenic. Glucose 6-phosphatase, remember, it is the only place within the body which contains glucose 6-phosphatase. Fatty acid synthase, naturally de novo synthesis is taking place in the liver. Carbamoyl phosphate synthase 1 is something which is related to urea cycle, and thereby not it will definitely happening in the liver, is the site for the synthesis of urea, and thereby it is present in the liver. So other than succinate, thiophorase. Let's go to the next question. The TCA cycle that forms a link with urea cycle. The TCA cycle intermediate which forms a link with urea cycle. Should have got the answer? It is fumarate. Now, how is it possible that fumarate? Remember, you have got in the urea cycle wherein ornithine combines with carbamoyl phosphate to form citrulline, and the citrulline has now entered into the cytosol. Citrulline combines with aspartate in the first stage to form argininosuccinate. Argininosuccinate is lyase and it releases fumarate. Fumarate is a link with the TCA cycle. Fumarate is converted to malate, malate to oxaloacetate, oxaloacetate to aspartate. So fumarate is converted to malate, malate is converted to oxaloacetate, and oxaloacetate is reconverted back to aspartate. So this link is got by fumarate. Fumarate, malate, oxaloacetate. These are the, but main link is fumarate, which links the urea cycle with the TCA cycle. That is why it is also called as urea cycle, TCA cycle, bicycle. So citrate, no. Citrate actually forms a link with fatty acid synthesis and TCA cycle. Isocitrate does not happen. Succinyl-CoA is also not the link. It forms a link with different amino acid metabolism, but not with the urea cycle. Let's go with the next question. A big question. Please take your time. This compares, in this question, there is comparison between carbamoyl phosphate synthase 1 and 2. So let's look at it. So all are true except. So you got your answer ready, you will be able to know. Right? All are true except this question. Both of them require ATP, whether it is in the mitochondria or in the cytosol. CPS1 is present in the mitochondria, yes. It is involved in urea cycle. CPS2 is present in the cytosol, yes, and that is how the compartmentalization is taking place. CPS1 is in the urea cycle and CPS2 in pyrimidine cycle. N-acetyl glutamate is the allosteric activator, yes, but not of CPS2. Ammonia is the source of nitrogen in CPS1, yes, and glutamine is the source of nitrogen in CPS2. Remember, the different sources of nitrogen are usually asked in different questions. What are the sources of nitrogen? CPS1 combines carbon dioxide with ammonia. Ammonia plus carbon dioxide to form carbamoyl phosphate. The enzyme is CPS1, and if it is CPS2, it combines glutamine, yes, or glutamine to plus carbon dioxide to form glutamate and carbamoyl phosphate. This is happening in pyrimidine biosynthesis. So this is happening in the cytosol, this is happening in the mitochondria. But both of them require ATP. So let's go to the next question. All of the following statements regarding insulin are true except human insulin is made up of 51 amino acids. There are 3 disulfide bonds in one molecule. It is released along with C-peptide. Its secretion is inhibited by glucose. It is degraded by insulinase. All are true except its secretion is increased by glucose. Whenever glucose increases through dietary absorption and glucose levels have increased, this blood, when it reaches the pancreatic beta cells, this glucose will enter, this is the pancreatic beta cell. Okay, when this glucose enters via blood tube, which is insulin independent. Glucose enters and immediately glucose is converted by glucokinase to glucose 6-phosphate, etc. But most importantly, what it does is it causes membrane depolarization by acting on certain potassium channels, and whatever insulin which is there in the stored vesicles of the pancreatic beta cells, all this are released. There will be exocytosis of insulin are released because of ATP potassium dependent ATP potassium ATPase will be acted. It causes membrane depolarization, and thereby the vesicles will come and fuse with the membranes and release insulin. So glucose sees to that insulin is released from the pancreatic beta cell. So its secretion is increased, actually, whatever is stored. Now, is it made up of 51 amino acid? Yes, it is made up of 51 amino acid. It has 3 disulfide bonds, one within and some within the molecule itself. They are present. So it has 3 disulfide bonds in one molecule. It is released along with the connecting peptide. There is a connecting peptide. Along with this, it is released, but then this connecting peptide is cleaved off, and the C-peptide actually levels can be measured in the blood to know how much of insulin is being released. Instead of just measuring the insulin levels, we usually go for C-peptide. Why? Because C-peptide has a bit longer half-life when compared to insulin. So it gives a more accurate measure of insulin release. Why? Because insulin, after it is synthesized, it has got a short half-life. It will be degraded by insulinase. So we can't come to know the actual release of insulin. So C-peptide levels are measured nowadays to know how much of insulin levels are being, to correlate with the insulin levels. C-peptide, for every molecule of insulin that is released, this of C-peptide is also released, and so it will correlate properly with the amount of insulin. So because rest of insulin is degraded immediately very fast by insulinase. Let's go to the next question. All the following are true regarding metabolic effects of insulin on carbohydrate metabolism, except. So again, we are looking at an integrated question in which we are talking of insulin metabolic effects. This is something which should be known. It inhibits HMP shunt pathway. No, insulin does not inhibit HMP shunt. It also activates HMP shunt pathway also. It increases glycogenesis in liver. Yes, whenever in the well-fed state, glucose, whatever extra is there, has to be stored. So glycogen synthesis has to take place. So glycogenesis is increased in the liver. It increases glycolysis in all tissues. That is the mechanism by which it decreases the blood glucose level. It sees to that when glucose is adequate, it sees to that all the tissues can use glucose, especially in the extrahepatic tissues like the skeletal muscle, cardiac muscle, adipose tissue, where it will see to that more and more glucose transporter 4 go to the cell membrane, and more glucose enters into the cell, and glycolysis is the preferred pathway in all the tissues due to insulin. It inhibits glycogenolysis. Yes, when we have enough stores happening, why do you want, when enough stores are there, why do you want to release glucose from glycogen? So this prevents futile cycles. It inhibits gluconeogenesis. Again, why does the body want to release glucose when already enough glucose has come from the diet? So it does not. It inhibits gluconeogenesis. It inhibits HMP shunt. No, actually during all this process, it sees to that the HMP shunt is also activated, so that enough reducing bioequivalents are also there, and fatty acid synthesis also can take place during when in the well-fed state. So it will not inhibit the HMP shunt. So let's go to the next question. Insulin insensitive glucose transporters are present in all of the following tissues except erythrocytes, lens of the eye, cornea, adipose tissue, liver. You should have got this answer. The answer is adipose tissue. Rest all of them, all the tissues which have been listed out there do not depend upon insulin for the entry of glucose. Erythrocyte always use glucose irrespective of whether insulin is there or not there. Erythrocyte RBCs are exclusively, solely dependent upon glucose. So in a diabetic, insulin is deficient, but the RBCs are not going to be lysed because the RBCs do not require insulin for the entry of glucose. Lens of the eye, yes, they also do not require, because the lens are constantly keep on using, they are devoid of mitochondria. Lens are devoid of mitochondria. Why? Minute mitochondria is present in the cell, it causes light diffraction and we would not have been able to see it. So the lens of the eye is devoid of mitochondria. It does not contain mitochondria, so it is also solely dependent on glucose. So whether insulin is there or not there, well-fed state, fasting state or starvation, the lens is going to use glucose. So irrespective of the hormone balance within the body, it is going to use glucose. So it is insensitive. Glucose transporters are insensitive. Cornea also the same way, for prevent light diffraction, does not contain mitochondria and thereby it allows entry of light. Adipose tissue, no. Adipose tissue is dependent on insulin. So the glucose transporter 4, which is present in adipose tissue and skeletal muscle, these are translocated in response to insulin. So these glucose transporters 4 are insulin sensitive. Liver, again, liver cannot be dependent on insulin for its action. Again, it is glucose transporter 2 which is present in GLUT2 and this is also insulin insensitive. The brain also does not depend upon insulin for its for the entry of glucose. Otherwise, the diabetic the brain could not have been able to use it at all. But except the satiety center. Except the satiety center, which is insulin dependent, rest all are insulin independent, insulin insensitive. So liver, brain, erythrocyte, lens of the eye, cornea, all these do not require insulin for the transport of glucose into their cells and utilize glucose. Let's go to the next question. All the following statements regarding metabolic effects of glucagon are true except, again you got glucagon. So there are a lot of tissues and lot of pathways here. Everywhere in each choice there is one tissue and one pathway. So let's see how you have done it. So your answer is, it stimulates glycogenolysis in muscle. So if I had stopped at, if the question was it stimulates glycogenolysis, it could have been true. Yes, metabolic effect of glucagon is true. It stimulates glycogenolysis, but not in the muscle. It stimulates glycogenolysis in the liver only and not in the muscle. In the muscle, what stimulates glycogenolysis? Epinephrine. Why? Because glycogenolysis in the muscle, the purpose is different. The purpose of glycogenolysis, that is release of glucose from glycogen, when it has to take place, when the muscles require energy. And when do muscles require energy? When it is exercising, strenuous exercise. And during strenuous exercise, which hormone is released? Epinephrine or any of the other hormones. So epinephrine, what does it do? It stimulates glycogenolysis. Glucagon, what is the main function of glucagon? To maintain the blood glucose level. Can muscle glycogen contribute to blood glucose level? No. So naturally, glucagon will not have any effect on the glycogenolysis of the muscle. So this will be the false statement. Rest all are true. It stimulates gluconeogenesis in liver. Yes, gluconeogenesis takes place in the liver and the kidney, and glucagon increases gluconeogenesis. So it stimulates gluconeogenesis in liver. It stimulates lipolysis in adipose tissue. Yes, lipolysis, again fatty acids and glycerol are released by lipolysis, and this lipolysis is responsible for maintaining these fatty acids. Beta oxidation gives energy, and this more of acetyl-CoA forms ketone bodies and ketogenesis takes place. It stimulates ketogenesis in liver. Yes. It inhibits cholesterol synthesis. Yes. Glucagon overall inhibits cholesterol synthesis. So rest all are true except it stimulates glycogenolysis in muscle. So let's go to the next question. All of the following are high energy compounds except ADP, creatinine, succinyl-CoA, phosphoenol pyruvate, 1,3-bis phosphoglycerate. So your answer should be creatinine. Why? Remember, what are high energy compounds? High energy compounds, we always write it with a squiggle bond, the high energy bonds. So what are these high energy compounds? These are compounds which release more ATP, more negative ATP than minus 7.3 kilocalories per minus 7.3. If it releases more energy than minus 7.3, we call it as a high energy compound, more negative than minus 7.3. That is the hydrolysis of ATP. ATP to ADP minus 7.3. So rest all of them. ADP is also a high energy compound, whereas AMP is not. creatinine is not a high energy compound, whereas creatine phosphate is a high energy compound. From where did creatinine come? It came from the anhydrous cyclization of creatine. Phosphate is converted to creatinine. So, minute it is converted to creatinine, it is not a high energy compound. Succinyl-CoA is a high energy compound. Substrate level phosphorylation is taking place. All the CoA are high energy compounds. So, succinyl-CoA is also a high energy compound. Phosphoenol pyruvate again is substrate level phosphorylation. 1,3-bis phosphoglycerate, again their NADH is released, so it is also a high energy compound. It has more energy to phosphate. So compared to all this, it is creatinine which is the appropriate answer. Let's go to the next question. A vitamin required in the electron transport chain, ETC. You should be able to answer this. The most appropriate among all this is riboflavin. FAD, FMN both are present in the electron transport chain. NADH dehydrogenase and succinate dehydrogenase, complex 1 and complex 3. Vitamin A is not required. B12, B6, ascorbic acid directly not involved with the electron transport chain. They may be involved in some other manner like ascorbic acid, but directly not involved, required in the ETC. Direct requirement is of riboflavin, FMN and FAD. So with this we come to the end of this section of MCQs. Let us talk more other MCQs in the next session. Thank you.